Hello students, once again welcome you all in my YouTube channel Pacific Chemistry classes. So my today's topic is hydrolytic enzymes. They are also known as hydrolysis. It is a very important class of enzymes. These enzymes catalyze the hydrolysis of chemical bond, whether it is a type of our CC bond, it is a type of our NH bond, or it is a type of our phosphorus nitrogen bonds. So hydrolytic enzymes is an important class of our enzymes. As we know that what are enzymes? Enzymes are the biocatalyst and they catalyze different our biological reactions. Okay, so enzymes are basically classified into main 6 categories. First is our oxidoreductases, transferases, hydrolysis, lysis, isomerases, ligases. So these are the important classes of our enzymes, and hydrolysis is an important category of our enzymes. So we will talk today in the detail of hydrolytic enzymes. So basically they are present inside the lysosomes, okay, of our body. So this is the structure of our lysosomes, and there you can see this orange color are our these are hydrolytic enzymes, and this is the membrane of our lysosomes, and there are small transport protein, because we know that for doing all the biological process enzymes plays a vital role. So just introduction, hydrolytic enzymes are the enzymes which catalyzes the hydrolysis of the chemical bond. So hydro, name itself is hydro, no? So they will, with the help of our water, they will catalyze the reactions. They will break down the macromolecules into the smaller molecules. Example, our nucleic acids, esters, peptides. For example, if it is a protein, first protein will get degraded into the polypeptides, then they will further get divided into the amino acids and so on. Similarly, the polysaccharides, they will get break down into the disaccharides, and further they will get down into the monosaccharides. Example, lipids, like they will get break down into glycerol, then fatty acids and phosphate bases, etc. So this is the intro. Then lysosomes are the principal sites for the our intracellular digestions. They contain 40 types of hydrolytic enzymes. Main enzymes are proteases, nucleases, glycosidases, lipases, your sulfates, your phospholipases, phosphatases, etc. So all these names you can see in the suffix it will come ases. I will just discuss the name and nomenclature also. So generally what they are saying that 40 types of our these hydrolytic enzymes are present in our lysosomes, because all the intercellular digestion is taking place there in the lysosomes. So the important ones are these three. So these hydrolytic enzymes play a crucial roles in the treatment of the brain tumor also. So they have a lot of application in the treatment of our various diseases in our body. In general, hydrolytic enzyme split the different groups of the biomolecules. Means the main role of this enzyme is to break down, split the macromolecules into the smaller molecules. Now let us see the basic reaction of our hydrolytic enzymes. As we told that, as the name itself, hydrolytic means with the help of water they will break down the macromolecules into the simple molecules. So here suppose if it is a substrate like small molecule AB, with the help of water and with the help of our hydrolytic enzyme, what they will do, the A and B will get break down into AB. So just ionization of water also, H plus and OH minus. So OH will get attached with our A and B will get attached with the H part. So just you can see this is a simple representation of a hydrolytic enzyme mechanism. In biochemistry, hydrolytic enzyme is the largest and the most diverse class of enzymes. They catalyze the hydrolytic cleavage of carbon-oxygen, carbon-nitrogen, carbon-carbon, carbon-phosphate bonds, etc. So in overall biochemistry, there are lots of enzymes. So this is a very important hydrolytic enzymes has a role, because they mostly do the cleavage of all the types of the bond which are present in our body, like carbon-oxygen, nitrogen, carbon-carbon, carbon. We know that in our nucleic acids, protein, amino acids, carbohydrates, fats, all these bonds are somewhere present. So the nomenclature generally we know that substrate plus hydrolysis means first the substrate name will come, then hydrolysis. So substrate, for example, if it is nucleic acid is your substrate, so what you will take, nuclear and ase for the hydrolase part. So the name will be like this, substrate plus ase. So the Nuclease refers to an enzyme that hydrolyzes the nucleic acid. So what is the significance of the hydrolytic enzyme? As we know that they have lot of importance as they are present in the body of plants, animals and the human beings. So all the living organisms, they are present. So they cleavage the large molecule into the small fragments. They are used for the synthesis like excretion, sources of carbon, for the production of energy. Like means if we have eaten the food, so if it is not digested, how it will create the energy? It is how it is excreted, how the our food will circulate. Okay, so everywhere in the body of plants and animals, these enzymes are doing their own roles in the transport, excretion, regulation and our signaling process. They are specific in nature, means they will act on a particular enzyme, the particular part. Now just we will see the classification of the hydrolytic enzyme. So as the classification, if we talk about, so as the enzymes are classified into three main categories. So they are then you will read in the books or text, it is written EC, enzyme classes, point 1, point 2, point 3. Just I have written here na, EC1, EC2. So this is EC3 category we are talking about. So it may be written like 0.3.1. So your hydrolysis are further classified as hydrolyzing ester bond. So example of this is lipase, hydrolyzing glycosidic linkage, hydrolyzing ether bond, hydrolyzing peptide bond, hydrolyzing the carbon nitrogen bond, hydrolyzing acid anhydrates, and hydrolyzing the carbon-carbon bond. So in every category, some of the examples I have written. So now what is important, as we know that there are at least 200 categories of these enzymes. So what are the most important? I have taken 3. Here is esterases, carbohydrases and nitrogen containing compounds. So here we can say first is lipase. Okay, so they will act upon the substrate glycerides, as we see na. So when they are acting on the substrate glycerides, so what will they form? They will produce the glycerol and fatty acids on the hydrolysis product. Means lipase enzyme, the substrate is glycerides, and this substrate will get hydrolyzes into glycerol and fatty acids. Similarly, carbohydrases, it's our example of this type of the category is fructosidase enzyme. Its substrate is sucrose, means it will get converted, this enzyme will act on sucrose. So this is a substrate, this is enzyme, this is the substrate, and it will convert this substrate into the simpler product, that is the fructose and glucose. Okay, so this is the hydrolysis product. Similarly, third category, nitrogen containing compound. So the enzyme is proteinases. Proteinases will act upon the substrate protein, and this substrate will get hydrolysis into polypeptides. So this one example you can learn. This is enzyme lipase, substrate is glycerides. Enzyme fructosidase, substrate is sucrose, will get hydrolyzes into fructose and glucose. So second example of these 3 categories, second is our lecithinase. Lecithinase is our enzyme. This enzyme will act on the substrate lecithin, and it will get converted into choline and phosphoric acid and our fats. So similarly here second example is alpha glucosidase enzyme. It will act on the substrate maltose. It will get hydrolysis into glucose. Okay, then next example of this category, proteinases. Here protein containing compound ureases is an enzyme. It will act upon the urea and it will get converted to urea plus amino acids. So these are the 2 examples, lipase, your lecithinase, and third example of this category is phosphatases, pectin esterases. So here I have written 5 examples. You can check them. Like we have amylase, beta glucosidase, beta galactosidase and cellulose. Amylase will act upon the starch and it will convert into glucose. Beta glucosidase will act upon the cellulose, and galactosidase will act upon the lactose. So here is spelling, you can understand cellulose, lactose. So they will act upon the cellulose, they will act upon the lactose, milk, and they will act upon the cellulose itself. So here this category polypeptidases, asparginases and deaminases, means they will break down the amino acids, they will act upon the aspartic acids plus your byproduct will be released. So this classification anyone you can write as per your comfort. Okay, so you can take the screenshot of this. Now the application of the hydrolytic enzymes. Hydrolytic enzymes have a vast number of industrial applications, like 75% mostly they are used in the industrial manufacturing process. So generally they are used for the waste treatment, as our waste is macromolecules of polymers and other things, so they are used to degrade the waste. They are used in the textile designing, they are used in the dairy industries and milk and milk products, foods and feeds, ripening of fruits, they causes the food spoilage. So they are microbial products formation, they are used mostly. They are used in the detergent industries, and in the detergent industry mostly our proteases is used. They are used in the leather industries, they are used in the paper and the pulp industries, they are used for making the biofuels, biocompost and all. They are used in the baking industries, cosmetic industries. So I hope you like the video. You can take the screenshots and complete your notes. Thanks for watching the video. Do share with your friends.